Okay, good afternoon, everybody. We're really glad that you were able to make it today. We're excited to have this little Q&A forum about COVID. I know there's a lot of questions and everything out there. I'm Dr. Goffett. Some of y'all may know me. If not, hi. I am the parasitologist on campus and just here to kind of help make sure everyone has all the correct information about COVID.
So I'm Dr. Fran Lemoyne. So the department I teach, in my area of expertise, cell biology and virology. And so I'm happy to share my knowledge with all of you and hopefully answer any questions that you might have about the virus, the vaccine, and anything in between.
My name is Lima. I'm a professor in the microbiology department within the School of Physical and Biological Science. If you have any questions, I will try my best to see if I can answer your questions.
Hi. Anybody have any questions that we get started with, or?
Do you know if there are any long-term effects of the vaccine?
So, okay, so let's take a history of vaccine development, and we'll talk about what makes this vaccine different than other kinds of vaccines that have been developed, right? Okay, did you guys hear me pretty well? Okay. So, dating back, there's historical evidence dating back to the 11th century of the first sort of human attempts at vaccination. It was called variolation, where they would actually take a little bit of smallpox and scratch it onto people's skin to give them smallpox, right? That's a terrible vaccine, right? Because you're actually giving the person the thing that you don't want them to get. But the way that variolation worked is you give them a very small dose and hope that they don't get sick and instead have a very mild symptom that eventually would allow them to be protected if they ever came in contact with a real high level of infectious agent. So that was sort of the standard of care for vaccination, and only for this one particular type of infection, smallpox, which was a big deal, right? All the way up through the mid-1700s. This is when it was brought to England and it became pretty standard practice. So later, in the late 1700s, they improved upon that process. And there were scientists who recognized that, well, there are some people who never get smallpox. Like in an outbreak in a particular village, there were people who didn't specific occupations, right? Milkmaids, these women who would milk cows. Everybody else around them would get sick. They would develop some. Milkmaids never got sick, right? And so what's the first rule of science? Make an observation, right? So that was the observation. Milkmaids never got sick. Why? Why did they not get smallpox infections? Well, they realized that they had a different kind of pox infection called cowpox. So now the light bulb went off. We don't have to give people a little bit of smallpox to make them protected against the smallpox virus. We can give them cowpox instead. And so they started injecting people with cowpox. And that would create a very mild type of infection, but it was still an infection. Still not a good kind of vaccine, right? But it was better than giving them a little bit of smallpox. So eventually, we improved on that process even more. Now we weren't giving a different type of virus to protect people. Now we recognize that in the lab, we could sort of grow these viruses on cells, and we could select through a process of artificial selection, right? In the lab, we can generate evolution to create a version of the virus that is weakened or specifically able to infect a different type of cell. And we refer to those types of vaccines as attenuated. So imagine if you had a virus that was really good at infecting human cells. And in the lab, we take that virus and we keep passaging it on monkey cells over and over and over and over and over again. And after 15 generations, we have, through the course of selection, artificial selection, created a virus that's really good at now infecting monkey cells, but maybe not so good at infecting human cells anymore. It's still alive, and it's still the same virus, but we have selected mutations that make it less infectious. And they then inject that into people. That's still a live vaccine, right? Even though it's now attenuated, it's still a live vaccine. So there are some dangers with that. It could mutate back in the person that you injected, and they could develop a strong infection. You're still causing an infection in that individual to stimulate the process of immunity. So again, how can we learn to improve on that process? Now, we don't want to give live vaccines anymore. That's not as safe as we want it to be. So we discovered, how can we tease apart the piece of a bacteria or the piece of a virus that actually is something the immune system is going to see? So instead of using a whole virus or a whole bacteria, we can pick one protein, which is on the outside of that virus or bacteria, and inject that into a person. One protein is not going to cause that person to get sick, but it's enough to stimulate the immune system to think that they've come in contact with a pathogen, and that causes immunity. So we call those types of vaccines subunit vaccines, right? When you inject a protein or a part or a piece of the bacteria or the virus. Subunit vaccines were great, and we developed lots of subunit vaccines for many different pathogens. The problem was you had to find the right protein to use, and proteins, as you learned in your classes, have to be in the right condition for them to keep their specific tertiary or quaternary structure. So we have to make sure that the temperature and the pH and the salinity, everything is just right. And to do that, we had to add lots of preservatives to those proteins before we could inject it into people. That's not good, right? We don't want to eat foods with a bunch of preservatives. We certainly don't want to inject it into our bodies. So now, about 2 years or 20 years ago, we started to expand and improve on that process even more. We don't need to inject a protein into a person. We can inject a messenger RNA that's going to code for that protein. And messenger RNAs don't require all the same level of preservatives that the proteins do. The other advantage is that the messenger RNA vaccine can be very easily swapped out. That sequence, that coding sequence or open reading frame can be swapped out really quickly like Legos, and we can modify it to be unique to a particular virus. So right now, we're in a stage of the first worldwide effort at vaccination, period. The first time we've ever had a worldwide attempt at vaccination, and it happens to be a messenger RNA vaccine, which has a lot of people concerned. Okay, so why are we going through this big history? There has never been a long-term effect for any subunit or non-live vaccine ever. When people have had reactions to vaccines, those reactions happen from 15 minutes to a week, and it can vary significantly depending upon the person and depending upon what that vaccine might have been. But there is no evidence in any way, shape, or form that there is going to be any long-term effects. But we know that there are potential long-term effects to having an infection, right? Specifically, these coronavirus infections can have some very serious long-term impacts. So that was way more than she actually wanted in response to that question.
Actually, I want to ask you, what specific long-term effects are you worried about in person?
It was just a general question. There's just a lot of rumors going around about like infertility and all of that. So I just wanted to hear y'all's opinion on it.
Right.
So the big thing about the rumors that started about it potentially causing fertility issues is because there is a small bit of similarity between the S protein, the spike protein that the messenger RNA encodes, and a placental protein. And so there was an idea, right? Not a good idea, but someone had the idea that that similarity would be enough that you could make antibodies against this placental protein, and that could ruin the chances of you having a successful pregnancy.
Okay.
That similarity is insignificant. We start talking about similarities, right? For those of you that are in your freshman biology classes, we talk about the differences between organisms, right? How similar are we to chimpanzees, right? So that's the level of similarity that we're talking about. So it's not significant, and that there is no evidence that there would be any type of cross-reaction to it.
And on top of that, if you look at the virus, so generally speaking, how does a virus get into the cells? First of all, a virus must go into the cells in order to increase its numbers, right? As a population, your ultimate goal is to pass over genetic materials from one generation into the next generation in the same time to increase in numbers. Every single species is doing exactly the same thing, right? So the idea is a virus must find a suitable cell type, a perfect cell type. Not every single cell can house the virus, right? So how can the virus know? The virus is blind, the virus don't have hands, so how can it know this is the right cell that I need to go in and I can take control of the cell and I can make more copies of myself? That is via a viral protein and a host surface protein in that specific interaction. And guess what? For this COVID-19 or SARS-CoV-2 virus, its most desired target is called ACE2. And guess what? ACE2, this is a host surface protein. That is a host surface protein you can find in many cell types, in many organs, including our own reproductive system. In other words, if we give a chance for the virus to get into our reproductive system, they will recognize our cells there and they will take control and ultimately probably killing our cells within the reproductive system. So if we really have to worry about infertility, we probably need to worry about the virus, not the vaccine. Because the virus, in theory, is able to attack the reproductive system because of the existence of these ACE2 receptors, just in theory. And that's also why, because of this wide distribution of ACE receptors, that's why there's no one common, most frequent phenotype of the viral infection. Somebody getting virus may just have runny nose, somebody may have a fever, somebody else may have a diarrhea, and guess what? They are all contracted with the same virus. And you might be wondering, why not the same symptoms? Because of the virus get interacted with host cells at different body spots. Because different body spots, guess what? Have abundance of ACE receptors. Therefore, virus go in there and settle down there, depending upon where the first initial interaction site is, that's where your symptoms is.
So I actually want to add something to that. So as you can see, I am pregnant, right? So I actually had COVID in January. So that was before I got pregnant. However, there are multiple papers. So of course, they're like, if you're trying to get pregnant, you wouldn't get vaccinated, right? One of the reasons why is because, like Dr. Moore and Dr. Ma said, we don't know what it's going to do to the placental protein. We didn't know what it was going to do to the unborn piece. However, there are papers out now. New England Journal of Medicine has published a paper saying the different times, first, second, third trimester, what's the percentages of live birth? Is there anything wrong with the fetus? The College of OBGYN now, they're actually recommending for pregnant women to get the vaccine. And it doesn't matter which trimester they get. I've known someone that got the vaccine before they were pregnant, She has a normal pregnancy, she's about to give birth. I've known 2 people that got the vaccine in their 2nd trimester, gave birth to perfectly fine babies. Right? So usually when there's something new, it's always gonna come out, oh, it's gonna be, I'm not gonna be able to have kids. And there is gonna be somebody that gets the vaccine and they're like, well, I'm not able to have kids. Well, what if you had that issue before? Right? So that's why there's always that misconception, but there are papers now to back it up. So if someone's like, I'm not doing it because of infertility issues, there's Harvard studies, New England Journal of Medicine, and the Association of OB/GYN. There says there's absolutely nothing. It's not hurting babies, it's not hurting mothers, and they've done fertility treatment for individuals that have to go through IVF. They've had perfect pregnancies, nothing wrong with it. Right? So I have, personal vendetta in it. I've read a lot of papers, but it's not affecting fertility. If there is something wrong with your fertility, you got the vaccine, maybe it was before the vaccine. Right? But a lot of people like to blame vaccines because they're like, oh, this is new in my body. This did it to me. What if you had issues before? Right? There's plenty of papers out there that you can read, and it's not messing with babies, it's not messing with moms. So there's plenty of papers to justify it now.
And I can attest to that too. So I got my first vaccine at 2nd trimester, and then my 2nd vaccine was in the 3rd trimester, and my 4-month-old is perfect.
We have a
As
question.
long as she's not biased, it's fine. And the other thing with that, most of the time people think about long-term effects or side effects associated with vaccination. Those things only occur when you have a continuous buildup of something that's toxic in the body, right? The great thing about a vaccine, specifically the great thing about these mRNA vaccines, is the messenger RNA is completely undetectable 72 hours after inoculation. So 3 days after the process of receiving your vaccination, the messenger RNA is not there anymore, and your body has started to develop those antibodies that are specific against that protein, the S protein, that the mRNA cause the synthesis of in the cells. But drugs that you take, right? Like let's say you take a specific medication for, high blood pressure or, heart disease. You continue to take those medications as maintenance medication, and that will continue to build up in your body. Every time you take another dose, it keeps those levels high. We need that because that's the purpose, right? That's what it's serving. Vaccinations don't stay in your body for a long period of time. So there's no chance for that long-term infection.
Go ahead.
So I have a friend who refuses to get the vaccine because he said that it usually takes years for them to develop a perfect vaccine. So he thinks this one, something must be wrong with it since they came out with it so quickly. Is there anything wrong? Like, how do I counteract that? Because I think.
Yeah, it's really hard because there's so much misinformation out there. And most people think that scientists didn't start developing the vaccine against coronavirus until December 2019. That's not true. This vaccine development actually began in 2001 or 2002.
2002.
And so in that process, remember I told you, this is decades long. Scientists recognized that the possibility of these mRNA vaccines was where we needed to go in the next generation of vaccine development. So at this point, the development of this process is what's going to allow us from this point forward to make vaccines at a much faster pace. The part that's limiting is how do you figure out what's the protein of the virus or the bacteria that you want to make antibodies against. That's the hardest part, is to figure out what on that outside of that shell, that protein shell, is what you need to make antibodies against. Once you have that, it's a snap, right? So what I have likened this to, if I asked you today, right, your charge now is to go and design a brand new type of airplane. You're not starting from the same place that the Wright brothers started in the design of an airplane, right? You have all of this information, all of the successes and failures of all of these other very fantastic people who have created a process We understand things better. So of course you're going to be able to do every step from this point faster, right? And then I was explaining this to a woman that works across the hall from me, and she said, okay, I'm going to go and talk to my mom about this, little 84-year-old woman. She's trying to explain to her why the vaccine was good. And she said, it's just like if today I want to make a strawberry cake and tomorrow I want to make a lemon cake. The recipe is the same, except you add strawberries instead of lemons, right? So once we create the cake recipe, once we create the ability of the mRNA vaccine to be efficiently delivered to our cells, we're just swapping out one piece for another. And from that point forward, we're going to be able to make vaccines much faster.
I would just add to that a lot of the time, because in the past it took about 10 years or so to develop a vaccine, but the science part doesn't actually move that slow. It's getting the funding, getting volunteers, all that sort of thing. And we do the steps of the trials one after the other. So this particular vaccine, money was no issue. Everybody was throwing money at it. People volunteered in groups, and they did all the different steps kind of at the same time. So that was another thing that kind of helped speed this up a little bit. So one of the things I hear is that we skip steps in the development of this vaccine. But that's not the case. It's just everything happened faster because it was so urgent. So that's another thing that this is about.
Thank you.
Oh, just hold on a second. I think Dr. made an excellent point, because if you look at the application for the approval of FDA approval, you have to go through trial 1 trial, stage 1 trial, stage 2 trial, stage 3 trial. All of the stages require volunteers, human beings, to be tested, not just animals, right? And not to mention, right now, with the pandemic going on, there are already people being infected. They are perfect population sample size to be recruited, because they want to be cured as well, right? So that thing itself accelerated the whole process a lot.
I thought about joining the vaccine trial just so I could maybe get the vaccine sooner. But I would have had to drive far away regularly to get the testing for the blood work on myself. But I considered it. So there are a lot of people who did volunteer for those trials for those same reasons.
And one quick thing from me, sorry. As Dr. Moss said earlier, this is SARS coronavirus 2. There was SARS coronavirus 1, that was a SARS outbreak in 2002. That was, everybody was like, you have SARS? I mean, I vividly remember that. It wasn't this bad, it wasn't this prevalent, but it was, yes. Everybody went, you have SARS? That was the first iteration. This is an extreme mutation. It's in the same basic family, if I understand correctly.
And then MERS also.
So it's the vaccine was started then. So it's a 20-year-old vaccine. It's not new. Somebody have a question?
If it's a 20-year-old vaccine and it's already developed, then why are they creating more variants to try to stop it from spreading more? If it should have already been taken care of.
So you say, why are they creating more? You mean
no, more variants. More.
Why are there variants?
Yes.
Okay.
The vaccine
So
working.
So it comes down to numbers, right? So when we think about, and I'm sorry if there's anybody in my virology class, because we just talked about this in class this week. When we think about the process of evolution, right? We think about a variant as a mutant, okay? So that means that one progeny is different than all of the other progeny, right? Think about that process of evolution and how long it takes, right? So in that process of evolution, it takes so much time to see a big change. I know if you haven't taken any classes with Dr. Aiken, one of his favorite examples is he talks about the evolution of a giraffe, right? Remember that, JD? So evolution of a giraffe. Giraffes started with necks that are this long, and then the trees got taller and taller and taller, and so a giraffe with a slightly longer neck, right? We all know tall kids in school, right? That giraffe was going to be able to eat more than the other giraffes. It ate more, it looked better, it got more mates, right? And so the kids of that giraffe, the progeny of that giraffe, were then going to have more numbers in the population. And so over time, you get more and more and more tall giraffes, right, long necks. That's the process of evolution. That happens, the driving force in that process of evolution has to do with how quickly you generate mutants. And some of those mutants are advantages, a taller giraffe, but some of them are going to be shorter giraffes, right? So the shorter giraffes aren't going to be able to eat, and they fall out of the gene pool. The taller giraffes with a longer neck are what creates the bigger group of the population later. The same process happens with viruses. The difference is the scope of the process. When we think about animals or plants, we're thinking about, how many progeny do you produce in one generation, right? So I always give this example in my genetics class. We think about the gestation period for an elephant is something ridiculous, like 2.5 years or something insane, right? So you would never do a genetic experiment on a giraffe, on an elephant, because it would take you so long to get the baby to try it, and then we all know numbers are important. So you'd have to repeat that experiment over and over and over again. Viruses replicate to extremely high numbers. At the height of an infection for a person who's infected with HIV, at its most robust level of infection, one single infected cell can produce 100 million HIV virions, that's babies, in one 24-hour period. 100 million kids going out. That's a lot of mutations that are happening in that area, and the one that has the advantage is the one that's going to infect the next cell and then go on and continue that in the gene pool. So when viruses replicate really quickly like that, and they expand to such high numbers, you're going to see mutants develop very quickly. And as those mutants develop, some of them are detrimental to the virus, and we never see them. But because we're talking the power of numbers, it's the one that has the advantage, able to infect at a higher rate or transmit from one person to the next. That's the one that we see in the next generation of infection. So the vaccines don't really have anything to do with the development of these mutants. It has to do with the genetics and the fast reproduction of the viruses. Now, what happens as these viruses, this virus in particular has an RNA genome, and RNA is much more unstable than DNA is, so they tend to accumulate mutations faster than viruses that are DNA genome viruses. So there are lots and lots of mutations in every virus that's produced, not just from one person to the next, but even within an individual during each round of infection. It's the one that's able to infect better because of what I call dumb, stupid, rotten luck. That mutation was better at infecting the next cell, and then it just goes out from there. So some of these mutants are so effective at transmitting from one person to the next because they're really good at infecting the cell. They get into a cell faster, which means they get out of the cell faster. That's what causes this expansion. More people are going to get that variant or mutant than others because those replicate slower. It's a tortoise and a hare, right? So what we have to do is try to pick, and we were talking about how to find the right protein to use in that vaccine. We have to find not only the right protein, but hopefully the protein that is the most stable in terms of the accumulation of those mutations, so that we're not picking an area that's constantly changing. Because when that area changes, the antibodies might not recognize it, and that's where we don't have the protection level. So that's one of the reasons why scientists are having so much difficulty creating a vaccine against HIV, because of the extreme fast replication rate, and the proteins that we want to use mutate very rapidly in a single round of infection. We're very fortunate in this case that coronavirus itself, this COVID-2, has an S protein that is relatively stable. The bulk of the protein stays the same, even through all of these different variants. What's different between the Delta variant and any of the other variants, right, the original Wuhan strain or the Alpha variant or any of these others, is that in the process of attachment and entry into these cells, right, Dr. Ma said it attaches to the ACE2 protein on the surface, and then it enters into the host cell by receptor-mediated endocytosis. So what happens, it goes into the cell, and it goes, you guys remember, it goes early endosome, late endosome, and lysosome, right? In the early endosome, there's a host, meaning our cells have an enzyme that chops one of the proteins so that we can have fusion and release of the genome inside the cell. The Delta variant doesn't need the host protease, the host protein enzyme to chop that off. It can do it automatically. So other variants, other mutants, have a slower round of infection because the Delta variant can get into the cell and it's off. So that's why it's replicating to much faster levels. And because of that heightened level of replication, it's going to have a higher level of transmission from one person to the next.
I think one of the other things that might be objective is he said it's a 20-year-old vaccine. The technology is 20 years old, but a vaccine that is aimed at SARS or MERS is not going to work on coronavirus because remember, just like you said, different ingredients. So the technology was there, it has been there. It's just building on it to make a vaccine that is functional for what we're dealing with now. So I think that may be the other part of the question.
And to add on, go ahead. And to add on this, I want to tell you a little bit about something called a genetic diversity. So if you look at all species on Earth, there has to be something called a genetic diversity existing within the species. That's a driving force for the species to be able to persist within evolution. And what is the genetic diversity? Genetic diversity basically simply means is among individuals within the population, there are some genetic differences. And these genetic differences can be as big as a lot of gene variations or as small as a few amino acid changes. So therefore, if you look at what is the pushing force, what is the mechanism that every single species on Earth is able to establish genetic diversity so that when a disaster comes in, then one of the type of the genetic variation can survive. So that's the ultimate goal of having or establishing a genetic diversity. But what's the driving force of it? Mutation. Mutation is the ultimate driving force for genetic diversity for all species. But of course, for microbes, they have something special, for example, horizontal gene transfer and a few other things. But for all species, consistently, mutation is the driving force, the commonly shared driving force to establish genetic diversity. And I used to tell people, if you just take a bacterial culture of about 5 milliliter in a test tube, and then you can say that all the possible viable mutations you can imagine it's existing there, just by random chance, because bacterial cells will generate mutations, just like Dr. Lamar suggested. They grow so fast, and in each generation they produce so many progenies, and then by chance, just simply by chance, they will generate a huge array of mutations, all the viable mutations. Of course, you will ask, why only viable mutations? Because if a mutation is non-viable, you don't even see it, they're dead, right, to begin with. So therefore, it's possible that these mutants of the viruses are not just created. They maybe have been there in nature already. It's just this current condition is in favor of their taking over of the population. It could be true. Also that too.
Another thing I wanted to add to this was they've done an excellent job explaining mutation and evolution. I just want to kind of add that where this mutation takes place and where all these mutants are developed is in a host. So the longer the virus circulates in our populations, the more chances we have of generating more mutants. So the very fact that our vaccination rates are not where we would like them to be is going to create conditions that make it more likely to generate more mutants. And those mutants may be less harmful than the current ones we have right now, but they may be more harmful. And we don't know which way that's going to go. So that's a really important thing I think for everyone to understand is that if you're vaccinated, then you have less of a chance of becoming infected, which means a mutation wouldn't have a chance to occur within you, right? And then it wouldn't have a chance to get out. So to kind of slow down the development of these mutants, we need to get our vaccination rates up to a certain level. Did that answer your question? Okay.
Our answers keep getting longer and longer. So it's not answering faster.
Well, we're professors. We're used to talking a lot without interruption.
Are there any other questions or comments, concerns, or anything?
So how are people who are vaccinated able to still get COVID?
So we have to talk a little bit about what happens in the immune system, right? Okay, so an mRNA vaccine, you're inoculated with that, and the mRNA enters into your cells. Your cell machinery makes this protein, and inside those cells, that protein gets chopped up into pieces and then displayed on the surface of that cell, right? So different parts and pieces of a protein. Remember, three-dimensional shape, right? It's really important. And then it gets displayed on the surface of your cell. So it's not a whole protein anymore. It's just different parts and pieces. We call each one of those parts and pieces an epitope, right? Now, in theory, your immune system is supposed to make, because of genetic diversity, an antibody that recognizes each one of those epitopes. But that doesn't always happen. Sometimes, let's say there's, for simplicity's sake, let's say there's 40 epitopes that get displayed from the S protein. You might only make 35 antibodies to 35 of those epitopes, right? So, because not all of the protein is available, right? You think about what happens. This S protein, the spike protein, is on the outside of the virus. It's not every single part and piece is available for an antibody to bind to it when it's in the virus itself, right? Because some parts are going to be on the inside, some parts might be touching the virus, and the antibody couldn't get up underneath it, right? So if that's the case, and one of your 35 antibodies was to an internal part of that protein, that antibody is almost useless, right? In an actual infection, a challenge, it wouldn't be a benefit to you. So what we hope for is that by numbers, we're going to essentially cover every single epitope that is valuable to have an antibody against, right? So if you get one inoculation, and that inoculation gives you coverage of 35 of the 40 epitopes, and by dumb, stupid, rotten luck, none of the 35 antibodies was to a valuable epitope. When you get exposed to coronavirus, you're going to get it, right? You're going to get COVID. That's the reason why we have a second dose, right? Because a second dose is going to give you a second chance to make more epitopes, because you don't cut in the same place each time, it's random, and you have a different chance. Let's say now this time you have another 40 epitopes and another random 35, but it's not necessarily the same 35 that you had before. So you are improving your coverage by having a second dose. This is exactly the same process and thinking of why you need this booster third dose to add additional coverage. So some people are getting infection because the type or the diversity of the antibodies that they produce wasn't enough coverage to protect against a coronavirus challenge. But what we do know is that even if they are infected, immunized, vaccinated people have a much reduced chance of a serious infection. They're experiencing much milder symptoms, which rarely leads to hospitalization or even mechanical ventilation. And that's been a tremendous advantage.
And also want to add on to this is because the vaccines right now are all targeting, regardless the DNA vaccine, MSCR, RNA vaccine, they are all targeting the spike protein, right? There has been some research suggesting that spike protein of this particular version of the coronavirus stays in a folded way. By folded, I mean just forget just like this. If I put my hands in the pocket and approaching to you, and then who knows, I'm gonna take out my wallet and give it to you, or just pull out a gun and shot you, right? Who knows? Nobody can be able to know. That's the same thing. If the spike protein stays folded, then the antibodies won't be able to recognize it because it stays folded. Then you will say, will it stay folded on forever? No, it's because when the virus is interacting with host proteins, the folding will be cleaved open so that the spike protein will be exposed, will be available, will be interacting with receptor proteins, the ACE2. But the problem is we don't know when it's going to happen, and we don't know how much time of the window this is going to happen, allowing not a receptor, antibodies to interact with it, to neutralize with it. It may be such a short window, it may be a long window, it's right? We don't know. So therefore, that's why in some cases, there are some vaccines not protective, but in other cases it is. It's just, in this case, this process is not completely controlled by a timing mechanism. It's random. It's random. So that contributes to why the sort of the leaking of the vaccine rate is it's not so pleasant for this one.
And you know, if we wanted to design, if the only thing we cared about in vaccine development was the creation of neutralizing antibodies, what we would do was give everybody coronavirus, right? Inject coronavirus into somebody, and I guarantee you, you're going to develop antibodies against it. That's not a good vaccine, right? Just like arsenic will kill every bacterial cell that it comes in contact with. It's a terrible antibiotic to take if you have a sinus infection, right? So the process of trying to protect people while saving their lives means that we have to try to cover as many bases as we can in terms of this randomness. And that is very difficult for people with limited scientific knowledge to understand, right? Because the question is always, why can't you get it right? I got the vaccine, why did I still get sick? You know, why did I still get the infection? And that's where we have a responsibility as scientists, and you have responsibility as students, because now you're learning more to share that information with other people. People in your family, your friends, right, your grandma, when they ask, you know, questions. This is where we have that responsibility to share with them. Because once people understand, even though it's a difficult concept, right? Nobody wants to hear it's random, but we can hedge it so that we have the results that we want at a much higher rate.
And kind of thinking about the efficacy of vaccines, a lot of the things that we use to keep ourselves safe are not 100% effective. Seatbelts aren't, birth control is not, but it still gives you much greater odds of having a positive outcome in that situation, right? So it's the same thing with the virus. You might still get infection, you might still get a little sick, but it's going to give you more protection than if you had nothing at all. So like definitely he's pointing out, it's a numbers game, it's an odds game. You know, what are the odds that the vaccine will protect you? Great, much greater than the odds of, you know, not having the vaccine.
Any other questions, comments, concerns? Anything you'd like our scientists to address? Okay. Do you guys have any closing comments?
You know, I have all sorts of people that I'm friends with on Facebook. People who I have known through my professional education, and people that I knew in kindergarten, who did not go to college, and, you know, and have different types of jobs than I do, right? And that's okay, right? We can be friends with all these people. The problem comes when you have a very difficult concept where people with certain knowledge can explain things on a different level, right? I told you, I feel a sense of responsibility to explain things to people. But I have all the time friends sending me text messages with a link to a school board meeting where someone is getting up and shouting about, you know, my kid can't wear a mask because they're going to get carbon dioxide poisoning, or, you know, any of these other things. And so I've been asked the question, how do you tell what's fake and what's real? So I think there's 3 criteria that you can use to tell what's fake and what's real. Number 1, scientists and medical professionals will never use the word vax. We always use the word vaccine or vaccination. So if you see that word vax somewhere, it's probably fake news. Scientists, medical professionals rarely cheer for each other. Okay? So if you are explaining your science, you're not going to have somebody standing, right? Dr. Dockett is not saying, yeah, go, tell them. You don't do that. I mean, you're not there, right? So when you see people at a science meeting and you hear the crowd shouting back and egging that person on, and that person is saying horrible things about other people's research or other people's ideas, that's not real. Scientists, medical professionals don't do that. And the third thing, if you see a physician or a researcher, some scientist, and they do not claim an affiliation with a research institution or a hospital or some type of institution of higher education, there's probably a reason, right? If they had to leave to create their own toxicology consulting firm, there's a reason why. So pay attention to responses of a crowd, pay attention to what they're saying, and pay attention to their affiliations. Listen to the people who are involved in treating people, not people who are involved in inciting panic, not people who are waiting for the cheers and the jeers behind them. Those are actors. And that misinformation is disgusting. This is what is separating our society right now. So use your rules of judgment and critical thinking to tease out the differences. It's hard when they're saying something you want to believe, right? Like a mask. Oh, that's such a horrible thing. If masks cause problems, we would have no surgeons who lived past 30 years old, right? They're wearing masks all day long. A neurosurgeon that does a surgery for 22, 24 hours at a time. How could they possibly stand that long with this toxic thing on their face, right? That idea that, oh, you're going to get carbon dioxide poisoning. What's the first thing I always think in my mind when I teach freshman biology? The one thing I want you to remember from this class is the concept of homeostasis, right? The concept that the carbon dioxide level or the pH level of your blood, we spend a tremendous amount of energy to make that constant as we can possibly make it. It doesn't matter. If you pumped CO2 into your body, we have a buffering system that allows that to be overcome. So use the knowledge that you have and help to educate other people on it.
I heard some really good advice a few weeks ago, and I really took it to heart. If you read a headline or an article and you feel a strong emotion towards it, good or bad, okay? When you strongly agree with it, you strongly disagree with it. That's your signal to take it with a huge grain of salt, right? Because you want to go find that's the point where you know you need to go look for other sources, look for other perspectives. find good, legitimate news sources and things like that. And so you can kind of make sure you're not getting caught up in the emotion of it, because sometimes our media is trying to get us to feel those strong emotions, right? Because that drives clicks and views and all that kind of stuff. So if you feel any kind of really strongly about it, news should be boring, and this is what I'm saying, right? So if it gets your blood pressure up, that's your signal to kind of like, okay, let me stop and take a step back, reevaluate, and look deeper into it.
So for me, I want to just make two simple comments. Number one is the messenger RNA technique inspired by the messenger RNA vaccine now has made a huge advancement in the treatment of cancer. That's first one. The great news, right? Cancer is much severe than a lot of other things and affecting more people. Number two, a big shout out to other types of vaccine. Yes, messenger RNA-based vaccine is great, but don't forget we have other types of vaccines. If you are super 100% afraid of the messenger RNA-based vaccine, there are still other types of vaccines, DNA-based vaccine, like Johnson & Johnson, AstraZeneca, or there is one that is, for some people, is safer, the subunit-based vaccine from Novartis. So in other words, there are other options available. Okay? If you just cannot deal with messenger RNA, there are other options.
And I know some of y'all didn't ask questions today. If you just don't feel comfortable asking your questions out loud, please feel free to email any of us. I'm sure that's okay, y'all. Speak for you. Just let us know, or if you leave and you have questions later, if something pops up in your mind, please reach out. We will be more than happy to answer your emails and to talk with you. I know from my point of view, I don't want y'all to leave confused, but I also know sometimes people are hesitant to ask questions in a public forum. I know as a student, I was not a hand raiser. I was a wait till after class, or so I definitely understand that. So if that's the case, please don't hesitate.